Hey guys, it's Mr. Kennedy. I'm just back with genetics of viruses and bacteria. It kind of goes right along what we've been talking about, and you'll see why whenever we get into recombination and genetic engineering. When we talk about a virus, we need to talk about the structure of the virus itself. I always like to use a bacteriophage as an example because a bacteriophage would be this one down here. A bacteriophage has a protein cap or coat around it, and then inside of it it has DNA in this case that it uses to inject into cells and cause them to create more bacteriophages. The word virus actually means poison in Latin, and if you think about it, that's what viruses do. They poison our system, so it kind of makes sense. Now, this protein coat protects the RNA or DNA that's inside of the virus and makes sure that it survives long enough to get inside your cell. All right. Now, there are basically two ways that viruses can reproduce. The first one is the lytic cycle. Now, the lytic cycle, there's a picture over here on the right. It basically it has an attachment. The virus attaches. It injects its DNA or RNA into your cell. You have hydrolyzation, which is the joining of the viral DNA to the host DNA or RNA, and then you have assembly of the virus and then release. Now, the thing about the lytic cycle is whenever the viruses are released from the cell, it causes death of the host cell. The second type is called the lysogenic cycle, and the key difference here is in the lysogenic cycle, it doesn't necessarily have to cause death. It incorporates its DNA and causes it to make more viruses, but the viruses might be able to leave without causing death to the cell. But the lysogenic cycle can actually take two pathways. It can turn eventually into a lytic cycle and cause the death of the cell. So it's just a combination of the two, I guess. Now, when we talk about RNA viruses, probably the most famous RNA virus is HIV. Human immunodeficiency virus, which causes AIDS. And the thing about RNA viruses, they actually transcribe DNA from RNA. They do a reverse of what we normally do. And it's really easy to remember if you think of that reverse, because what they use is reverse transcriptase. Reverse transcriptase actually takes RNA and turns it into DNA, which is kind of neat. The next thing we need to talk about are viroids and prions. Viroids are viruses that are found within plants, and they're like naked circular RNA that is injected into the plant, and it causes the plant to create enzymes that may stunt the plant's growth, etc. Prions are infectious proteins. It comes in and it changes normal proteins into that protein. You might have heard of mad cow disease before. It was caused by prions. When we talk about bacterial genetics, the DNA looks a little bit different than it does in us. In us, we think of it as being linear DNA. In bacteria, it's usually circular DNA. And we talk about a nucleoid, which is the region of bacteria where you have the DNA pack, because it's not found in a nucleus, because remember, bacteria are prokaryotes. And is formed in what we call plasmids. And you know what a plasmid is. A plasmid, whenever I refer to a plasmid, I'm just simply talking about circular DNA, a circle of DNA. Now, it looks down here, DNA, when it goes through DNA replication, you get two different copies. Bacteria often reproduce this way as well. They reproduce by a process called binary fission. It simply means that the bacteria grows, and when it gets large enough, it copies its DNA and it splits into two. This is asexual reproduction, so both bacteria will be identical to one another. Okay, when we talk about bacteria DNA transfer process, there's two words we need to know. We need to know transformation and transduction. Transformation we've already talked about in Griffith's experiment, where he did like the blender where he put the bacteriophage, the radioactive material, etc. Transformation is whenever a bacteria takes up foreign DNA. It's being transformed. Transduction is whenever a phage that carries bacterial genes from one host to another, it carries it from one place to another. It's called transduction. And The last thing that you need to know definition-wise is conjugation. Conjugation is the way that bacteria may reproduce asexually, and they actually will create a bridge, and you can see it down here in this picture. They'll actually create a bridge here between the two cells in which genetic material will be exchanged back and forth, so it can change the genetic makeup of the bacteria itself. Now, when we talk about the plasmids, remember I told you about plasmids, oftentimes you'll see it as little circular DNA. In this case, it's blue and black, and it's going to be used later on to modify these bacteria by adding plasmids to them. Now, bacteria, realize, are tiny organisms, and they need to respond very quickly to their environment. That's the only way that they can survive, right? So if they don't want to waste energy in any way, so if they have an excess of an enzyme, they stop the process of making that enzyme. And if they need an enzyme, like they found a new food source, and they really need an enzyme to metabolize it, then they start the production of that enzyme. So they're very good at turning on and off their mechanisms. And it's regulated, turning on and off. They can regulate this by a suppressor gene a lot of times. Now, let me explain. Bacteria usually group genes together in what's called an operon or an operon. I call it an operon. And in the operon, it is going to have a promoter region, it's going to have an operator region, and it's going to have a list of genes. The promoter region is simply the region where the RNA polymerase binds so it can make RNA. So you learn about RNA polymerase in the central dogma. This is kind of like the Tata box in my mind, but the promoter is where it binds. Okay, the operator is a DNA binding site, so it can use a repressor protein to repress this in case it doesn't want this sequence to go, and then all the genes behind it. And you can see it down here. Now, the repressor protein, what it does is when it binds to that operator, what it will do is it'll cause the RNA polymerase not to be able to go, so it stops transcription, which stops the whole process, right? So if we look here, here's what happens. The promoter is where an RNA polymerase joins. Nothing's hooked to the operator, so the RNA polymerase travels down and it reads each one of the genes, right? That's a normal flow. And as it reads down, it makes mRNA, which is going to make the enzymes. Now, if you got a repressor protein attached to the operator, when the RNA polymerase comes up, it's unable to attach. So therefore, you don't get any of these enzymes made that are being coded for by the gene. So that's how you can turn it on and off in this operon model. Now, there are basically two ideas of this. There's a repressor operon and an inducible operon. A repressible operon would be one that whenever you have excess, you're able to repress it and stop it. An inducible one would be whenever you don't have enough of something, it automatically starts. So anyway, this is just kind of a brief, quick overview of a lot of different things with bacteria and viruses. I hope it helps you later on, and I hope you guys have a wonderful day.